Remember, the hollow cell is the blastula or blastocyst, the single layer of trophoblast cells, and the inner cell mass. Until the syncytium appears, we just call it trophoblast.
What kind of cells are the inner cell mass?
The inner cell mass is going to form the amnion membrane and the embryo proper. That's what we'll look at second. So this is a uterine lumen here, and the stratum functionalis deep to the epithelium. Now that we have a syncytial trophoblast, the few cells we can call the single layer the cytotrophoblast. All right, before that, just trophoblast. The green tissue is the syncytial trophoblast. More and more cytotrophoblasts form through mitosis and add to the syncytium so it completely surrounds the thinking blastula. Now it's embedding blood vessels, the maternal blood vessels. So remember, it's going to send syncytium up into the blood vessels so that the blood is in direct contact with the fetal tissue. The lacuna join. And the lacuna are just the pools of spiral arteries that have opened up into a common space. All right, so we're going to come back and look at this step by step. And then we're going to look at the development. This is the inner cell mass, if someone was asking. So the amnion, the epiblast, and hypoblast are what develop from that inner cell mass. So I'm going to reverse it, and like I did on Friday, instead of having the blastula moving into the stratum functionalis in this direction, I'm going to move it down in this direction. So the uterine lumen, or cavity, will be up here. And it's closed over. Still have our simple columnar epithelium. And our trophoblast cells have multiplied. And started to fuse together. So now we have our syncytium. So these are now called syncytial trophoblasts. And that's all one structure. All these cells are fused together into one large amorphous mass. It's really difficult to call it a cell, but it's one massive cytoplasm. Once that starts to occur, the cells that remain are identified as cytotrophoblast cells. Cells with a cytotrophoblast. All right? And they will continue to multiply. They will stay a single layer, but they will continue to multiply and will send hordes of cytotrophoblasts down into the syncytial trophoblast finger-like structures. Some of these cytotrophoblast will develop into fetal connective tissue or mesoderm. I had a red pen here, wrong color. And so that's the fibroblasts and the developing blood vessels. So we call that combination of tissue the syncytial trophoblast, the cytotrophoblast, the mesoderm, the extraembryonic mesoderm that's forming all of this, we identified as the chorion. And remember, chorion was fetal tissue, and we had two different types of chorion. So as it continues to develop, here you can see the chorion is entirely around the blastula. This is a cross-section of one of those chorionic villi. Out here is the syncytium. Let me turn this overhead like that as well. Here, so out here is the syncytium. Here are the cytotrophoblasts. And this would be the trophoblast cells that have formed the mesoderm, the extraembryonic mesoderm. We have fetal connective tissue, fetal blood vessels that I will diagram in later as we're looking at a more mature placenta. And then as this grows farther, it presses up against the surface of the uterine lumen that it just subsided, embedded beneath. Then we see a difference in the development of the chorion. And the deeper chorion forms the chorionic villi still, while the superficial or surface chorion is less developed. And we have the chorionic villi and the chorionic levy. Okay, so that's kind of what I drew over on the board. So this continues to develop further. I'll leave it partially developed on this side. This continues to develop further, branching. So on. The cytotrophoblast and connective tissue continues down into each of these with the mesoderm. And so this area of chorion is identified as the chorionic villi. And this area up here that does not show that extensive branching is identified as the chorion levy. Remember, you can spell it like this, or you can spell it like that, meaning smooth chorion. This is about where we stopped. So I did introduce you to the words decidua. So fetal tissue is chorion, right? And maternal tissue is decidua. So the decidua is the stratum functionalis, the entire uterine lining. Remember, the endometrium has two layers. Stratum functionalis, it separates at menstruation and becomes the menses, and the underlying stratum basalis that replaces and regrows the stratum functionalis at every menstrual cycle. So that entire stratum functionalis undergoes an inflammatory reaction where the fibroblast cells swell up. Interestingly enough, even though it's identified as an inflammatory reaction, there's actually a suppression of the immune response. And so when we talk about the immune system, we talk about active T cells and B cells and helper T cells. There's another type of a T cell called a suppressor cell that suppresses the immune response, and there's those multiply. And so to allow this invasion of fetal proteins, half of the genes in this fetal cells and those trophoblast cells came from the sperm and would be recognized by the mother's immune system as foreign. And so to suppress their rejection, the immune suppressor cells influence masking, if you will, of their marker proteins. But this inflammatory reaction is called a decidual reaction. We have 3 locations of maternal stratum functionalis. Immediately deep to the chorionic villi. All right, this is where the lacuna, the spiral arteries are forming the lacuna and supplying the blood aspect of maternal tissue. So this would be the decidua basalis. It's a confusing name because it's formed of the stratum functionalis, not the stratum basalis. It's basalis because it's basal to the embryo. So it's from, all of the decidua is from stratum functionalis. Just below the uterine surface, where we have the chorion levy, the decidua is called the decidua capsularis. And this is especially noticeable as this developing tissue grows out into the lumen of the uterine cavity. So notice that the surface lining of the uterus over the implanted embryo, as the embryo grows larger, that surface lining maintains its integrity and the lumen gets smaller, but the embryo and fetus are always covered by that lining. It's like having an anthill or something underneath the ground, the whole dirt lifts up. And so as the fetus grows larger, it's still covered by that surface layer of the endometrium. And so it makes more sense to call this a capsularis because it's forming a cap as that fetal tissue grows outward. Everything else not involved with the embryo, so everything that's not directly associated with the chorion villi or the chorion levy, so the rest of the uterine lining forms the decidua parietalis. So here on this lower image, we have a fetus now, and so here we would have the decidua capsularis, decidua basalis, and then across the uterine lumen, all of the rest of this would be the decidua parietalis. So again, decidua parietalis, decidua capsularis, decidua basalis. Chorionic villi, chorion levy, just deep to the decidua capsularis. The amnion is part of the inner cell mass, so that's what we're going to talk about in a few minutes. But I want to diagram my image over there and look at the placenta tissue itself here. Okay, so I'll leave this image up on the board a bit longer. May I erase this? So here's the cervical canal and the cervix. And here's the uterine lumen. But our embryo fetus is growing, so it's growing into that lumen. This would be the stratum functionalis, which is decidua now. So at this point, this decidua is going to be the decidua parietalis. associated with the decidua capsularis is our chorion levy. So this maternal tissue here, this is the lumen. This is decidua capsularis. This would be the chorion levy. Syncytium with cytotrophoblast cells. Though there aren't as many as this because they get fewer and fewer as pregnancy goes on. And then we have, associated with our decidua basalis, highly branched chorionic villi. What I'm drawing is these branches are actually microscopic. So there would be these branches and then 10 more sets of branching, more. The chorion levy. Chorion villi or chorionic villi. Now this would now be within a pool of maternal blood. So what we're seeing here. The spiral arteries have been opened up, pulled together. The maternal blood would be bathing each of these villi. And then this maternal tissue down here would be decidua basalis. All of the decidua, remember, are still part of the stratum functionalis. So when the baby is born, the decidua is going to separate from the stratum basalis. So all of the placenta and decidua basalis, as well as all of this decidua capsularis, because it would have the stratum basalis deep to it as well, is going to separate and be delivered as the afterbirth, after the baby is born. So this is what I've drawn here. Now, we have the baby. I've left the baby out. And I've left the amniotic membrane, which we haven't described yet, but let me draw that in there. So I'll just put a simple baby. Is going to be connected to the placenta via the umbilical cord. Make that easier because the baby's facing up. We'll have to draw the umbilical cord going around. All right, the amniotic membrane, will describe its formation in a moment, is going to surround the umbilical cord cover the surface of the placenta and form a sac which encloses the fetus. Now, the baby needs oxygen and nutrients, and it needs to give off carbon dioxide and waste material. So 2 vessels are going to leave the baby via the umbilical cord, carrying the oxygenated blood, and travel across the surface of the placenta and send branches down into the chorionic villi, forming capillaries, fetal capillaries. Even though they're outside the fetus, they're identified as fetal capillaries, and they contain fetal blood cells because they will re-enter the fetus. And these 2 vessels are known as umbilical arteries. And there was an immediate question in my morning class, well, why are they blue and deoxygenated if they're umbilical arteries? Well, we haven't done our cardiovascular system yet. However, we identify vessels by their structure, not their content. And structure is determined by what I was about to say what it contains in that circular reason. Structure is determined by the force of the blood within it. So if blood is leaving the heart, it's under high pressure because the ventricles are contracting. Therefore, the blood vessel walls need more smooth muscle in order to resist completely blowing apart. So blood that leaves the heart and is under high pressure is identified as being carried in an artery. To return to the heart, pressure has to drop. So blood flowing into the heart is at its lowest pressure. At low pressure, it doesn't need such a thick vascular wall, and so we define that as a vein. Normally, blood being pumped out of our heart is oxygenated. It's carrying oxygen to the heart. It's carrying oxygen to our other organs and to our skeletal muscles. But where do our adult hearts pump blood to pick up oxygen? In the lungs. So leaving the heart, we have pulmonary arteries carrying deoxygenated blood to the lungs, and pulmonary veins carrying oxygenated blood back to the heart under low pressure, because it's returning to the heart. This is the other example of where we have arteries carrying deoxygenated blood. So leaving the fetus, being pumped by the fetal heart, leaving the fetus, we have umbilical arteries under higher pressure, pushing that blood out the umbilical cord and into the placenta. In the chorionic villi, it picks up oxygen and returns under low pressure as oxygenated blood in an umbilical vein. All right, so we're going to have 2 umbilical arteries and a single umbilical vein. And this is going to carry oxygenated blood. We'll see this when we do fetal circulation in the cardiovascular system. It's going to return oxygenated blood back to the baby's heart, which will then pump it to the rest of the body. So we have 1 umbilical vein. So when the umbilical cord is cut, the pediatrician or the nurse or the OBGYN will always check to see that there are 3 vessels. There's nothing they can do about it if there aren't, but at least they can look for other additional congenital problems.